Hello, this is Shashank Biswas, and I am doing a project on DNA nanostructures review. This is a review paper. I am doing this paper under Professor Manish K. Gupta in the course IT 468 Natural Computing at DICT. Introduction. Adrian Seaman was the first to pursue the field of DNA nanotechnology and perform successful experiments to come up with an entirely revolutionary field of technology which could open a never-ending field of applications. With the never-ending need for products in the current world to fulfill the demands and requirements of the ever-growing population, the scientific community need a revolution to give the world something which could provide the vast needs in a very small resource utilization. DNA could be the key to that. With the current level of developments in the DNA nanostructure advancements, the time could be near when every technology could be shrunk to a level that could be carried in hand, let it be an electronic, mechanical, or biological device. In this paper, we try to address the developments that took place in the past decade and a half in the field of DNA nanostructures. Siemens' original motivation was to create a 3D lattice for his crystallography studies. He got motivation from the woodcut depth model. Nano-designing can be approached in two ways. First, top-down approach. The top-down approach employs natural and man-made blocks that are cast, cut or machine processed into precise products, small or large ranging from circuits to jumbo jets. The methodologies to produce nanoparticles from large material require high-energy ball milling, solution chemistry, electro-explosion, sonication, mechanochemical processing, etching, and physical and chemical vapor deposition. The pattern structures that are grown by appropriate lithographic and ion implantation are cornerstone of silicon integrated chip technologies. Secondly, bottom-up approach. The bottom-up approach is manufacturing nanomaterial by changing and organizing individual atoms and molecules into specific configurations like processes performed by molecular machines in living organisms. The self-assembly lipid molecules into nanostructures like liposomes could be considered as classical demonstration of the bottom-up approach of constructing nanomaterials. One more attractive example of atom-by-atom assembly of nanostructures is the writing of the IBM logo by placing individual xenon atoms on a surface with an STM tip. Now, structural DNA nanotechnology from design to applications. It became possible to create these nanostructures by the help of two following properties: tile-based method and DNA origami method. Tile-based method is comparatively old and DNA origami method is new and more appropriate for construction of bigger and sophisticated structures. Adrian Seaman proposed the idea of DNA nanostructures in 1980s with the help of different arrangements. He created many complex DNA structures. He used arrangements like double crossover motif, DX tiles, two-dimensional lattices, triple crossovers, and 4x4 cross tiles. Now, applications of DNA nanostructures. DNA nanostructures serving as a platform for directed assembly of synthetic vaccines. For the targeted delivery of antigen to the antigen-presenting cells (APC), which also include macrophages, dendritic cells, and B cells. This is an important step in starting a good immune response. Earlier researches showed the size and shape, surface charge, also hydrophobicity, hydrophilicity, and receptor interactions by an antigen can affect its adoption by APC. Then the targets are processed and offered to T cells for activation. It is seen that co-localization of antigens and adjuvants inside the same APCs can amplify antigen presentation and T cell activation. At last, activated cells assist in differentiation of antigen-specific B cells and creation of antibodies which are specific to the target antigen. Now, DNA nanostructures as templates to assemble organic and inorganic material on them. On the left, organic material templated on DNA nanostructures, and on the right, inorganic material templated on DNA nanostructures. The common strategy for making a DNA protein conjugate pair is functionalizing DNA with biotin and binding it to streptavidin. The 4x4 cross tile is modified with biotin at the center of the tile, with streptavidin is added to the mixture and bound. Clear two-dimensional periodic streptavidin arrays are seen by AFM. In later developments, streptavidin was attached on linear TX tiles arrays using biotin. using biotin along one or both edges of the array. Streptavidin can also be templated on DNA origami structures with controlled locations. We can also template metal particles on DNA nanostructures in an organized manner. One approach can be linear TX style arrays are periodically modified with biotin. Gold particles conjugated with streptavidin are precisely positioned on the self-assembled linear array of TX tiles. Second approach could be gold binding peptide molecules are conjugated with 4 by 4 tiles. Further addition of 5 nanometer gold particles results in specific binding of gold particles on the peptide molecules at the specified locations. Now, applications of DNA nanostructures in biological applications. For starters, we can make a cellular signaling path with the help of DNA nanostructures. Signaling using transforming growth factor (TGF) is seen to be dependent on clustering of the receptor particles. By functionalizing the pattern, a DNA nanostructure with closely spaced peptide molecules which bind to TGF receptor, sensitivity of these cultured cells towards TGF was marginally increased. The authors also proposed and partially constructed a multivalent thrombolytic delivery system to measure and deliver human tissue plasminogen activator and plasminogen, two important molecules in thrombolytic pathway. Now, construction of semiconductor nanocrystal arrays of quantum dots using DNA. Many studies on conjugation of semiconductor nanocrystals with biomolecules have been done. These nano-sized conjugates are biocompatible and serve some advantages other than organic dyes and lanthanide probes. The emission wavelength of quantum dot nanocrystals can be regularly tuned by changing the dot size, and a common light source can be used for simultaneous excitation of all different size dots. The perfect optical properties make these dots ideal for urophoresis for multicolor and ultra-sensitive applications in nanobiotechnology and nano-level bioengineering. Now, DNA nanostructures and theranostic applications. We have already seen the unmatched property of self-assembly of DNA nanostructures and also nanostructures in different dimensions. Since biological properties of the DNA can lead to use of it in other fields of biology like therapy and diagnostic, it can be used as biosensor for such applications. For example, DNA tetrahedra, which is 3D structure which is rigid and has well-defined structures. When it is created with various probes, it provides different types of structures for biosensing. Such structures also improve target accessibility and sensitivity. Other properties like AT and GC pairing and structural features of nucleic acid, where single-strand DNA provides flexibility and double-strand DNA provides rigidity, makes it easier to create structures. Nowadays, technology like DNA origami also provides great help in creating structures. Now, illustration of DNA biosensors. Using the famous thiol gold chemistry and DNA hybridization on the gold surface, eDNA sensor was created. However, there were lots of factors to be controlled to make it work properly, like interstrand entanglement and electrostatic repulsion between strands. This was created by using property of structural switching of 2D DNA stem loop structure. There would be one target DNA to which our complementary 2D DNA hairpin structure will bind and will produce the electric charge, which will get measured with the help of the transducer. Just by changing the probe of the eDNA sensor, property of whole sensor can be modified. For example, ATP sensor was created just by using the structural switching of anti-ATP aptamer. Now, the delivery system using DNA nanostructures. It was observed that this tetrahedral nanostructures were non-toxic and more stable than linear DNA inside the cell. These tetrahedra were also capable of crossing cell membrane. By using this property, new type of drug delivery system of CpG oligonucleotides was created. In the research of in the research, they created tetrahedral DNA nanostructures for the delivery of siRNAs to cancer cell of mice. Efforts were also made to create efficient drug delivery system using logical functions. Research by Douglas gives the description of robotic DNA device which can sense the receptor. It contained the barrel-like container locked by two aptamers. They made the use of AND gate such that both the aptamers reaches the receptor, then only the barrel will release the cargo molecules. Such intelligent delivery system can lead biology to better levels. Now, SL2B aptamer and folic acid dual targeting DNA nanostructures to combat colorectal cancer. In research done by Pengchao Sun and his team, they constructed DNA tetrahedra and modified it with SL2B aptamer and folic acid. This SL2B can prevent the growth of cancer cells by interrupting 11 vascular endothelial growth force notch signaling pathways. There is another component Td which was small in size and entered the nucleus quickly. So this SL2B multimers were created with Td and different numbers of SL2B. Best among several combinations was one SL2B per Td, and it showed great anti-cancer characteristics as compared to SL2B. There was also another component DOX which was further enhanced by functionalization of SL2B and F-DOX intercalated TD modified with 2F and 2S, and it entered in the nucleus by the TD. This modification caused the HT29 cell inhibition at much lower DOX concentration. DNA nanostructures DNA nanostructures also have limitations. There are some crucial limitations on DNA nanoassembly based on the materials and chemical properties of DNA. It is easier to think of DNA as a textile material than a hard framework material like cloth and weave baskets compared to bricks and sticks. The reconfiguration and self-assembly of DNA depends upon its ability to form weak interactions like hydrogen bonds and pi stacking interactions. This is because these weak interactions are all what holds higher-order DNA structures together. DNA nanostructures are soft and delicate substances. Actually, bulk hydrated DNA nanostructures constitute like a soft gelatin. AFM images of DNA nanostructures show that it is easy to pick out structures which are folded, crumpled, or squashed or damaged and broken. The above distortion in the structure result for three-dimensional structure in solution to collapse to a 2D structure on surface. Still, all these limitations can also be an opportunity for researchers who want to participate in DNA nanostructures field. Now some concluding remarks. The DNA nanostructures and their applications since the advent of this technology led by Adrian Seaman has been an interesting field which holds a great potential. DNA nanostructures hold the ability of programmability and self-assembly, which is the most feasible thing at nanometer scale. After programming DNA, it can make the desired structure without outside interaction on its own by self-assembly. Other inorganic nanotechnology does not allow abilities like self-assembly, which make them rely on slow and very careful atom by atom or molecule by molecule assembly by the designer using very high precision instruments. The DNA nanotechnology has a very promising future in biomedical applications, templated assembly, and DNA theranostics. DNA templated assembly could be used for making nanomechanical devices or used to make some optical devices by assembling materials with optical properties on the templates. Thank you. This was all from my side.